There's this really comforting idea about our biology that I think most of us kind of cling to. It's the idea of the blueprint.
The architectural plan, the master document.
Exactly. We think of our DNA as this fixed, rigid set of instructions. You get the blueprint at conception, and that's it.
Right.
It dictates your eye color, your height, your risk for every disease. It just feels inevitable, set in stone.
It's a very common metaphor. And, it's useful for high school biology, for sure. It helps with the basics of inheritance.
Sure.
But when you actually stack up the research we have today, and we have a really interesting stack to get through, you realize the blueprint is, maybe a little too static.
It's misleading.
It is, because a blueprint implies that if you build the house twice, you get the exact same house.
Yeah, and biology just doesn't work that way.
Not at all. If you really want a metaphor that fits the modern science, our genetics are much more like a dynamic switchboard, or even better, like a complex machine learning algorithm that's constantly rewriting its own inputs based on what it experiences.
That is exactly where we are heading today. We're going to challenge this whole concept of genetic destiny. We're looking at 3 distinct sources that, on the surface, they seem totally different. But together, they tell this new, much more complicated story about life's code.
Right.
We're going to look at the hardware. The old school, gold standard tech used to read DNA, then the software, the epigenetics that tells the DNA what to do.
And finally, we're going to look at a brand new study, one involving artificial intelligence, that suggests we might be able to diagnose genetic errors without ever looking at the code at all.
Which is the part that really blew my mind. So our mission today is to peel back these layers. We have a classic review on epigenetics and human disease, a technical breakdown of Sanger sequencing, and this new paper on using machine learning for early diagnosis.
And the through line here is really complexity. We're moving from just reading the letters to understanding the context, and now to finding patterns in the chaos.
Okay, let's start with that context. The software. We're looking at this source, epigenetics and human disease. Now, I've heard the term epigenetics thrown around a lot, usually in wellness circles, green juice or meditation.
Right.
But strictly speaking, scientifically, what are we talking about?
So if we break the word down, epi literally means on top of. So on top of genetics. The source defines it formally as altered genetic information without changing the DNA sequence.
Okay, so if we stick with the computer analogy, the DNA sequence, the A's, C's, T's, and G's, that's the hardware. That's the hard drive.
Correct. And that hardware is identical in almost every single cell of your body. Your neuron has the exact same DNA as your liver cell.
But they look and act nothing alike.
Exactly. So why?
Because they're running different programs.
That is epigenetics. It's the software layer that tells the hardware. Okay, you're a brain cell, so turn on the neurotransmitter genes and turn off the liver enzyme genes.
It silences some parts and amplifies others.
It's the conductor of the orchestra.
Yes. And this isn't just about cells differentiating when we're embryos. This source opens with a story that honestly it blows a massive hole in the hole, it's all in your genes theory. The case of the monozygotic twins.
This is a classic case in the literature. And it's one of those medical mysteries that really forces you to pay attention.
Okay.
So you have identical twins, monozygotic. Genetically, they are clones. They came from the same fertilized egg.
Right.
And in this specific case, both twins carry the exact same genetic mutation for a disease called adrenoleukodystrophy, or ALD.
That's a heavy diagnosis.
It is. It's an X-linked disorder. It involves the breakdown of myelin, the protective sheath around your nerves in the brain.
So without myelin, the whole system just fails.
The nervous system can't function.
Yeah.
It's usually degenerative, very severe, and often fatal.
So you have 2 clones. Both have the mutation that causes the myelin to break down. If DNA is destiny, you'd expect both to get sick.
That's what the blueprint model would predict, absolutely. But that's not what happened.
What happened?
One twin was blind and suffered from severe neurological issues.
Okay.
The other twin, completely healthy. No symptoms at all.
That is wild to me. The same DNA, same mutation. Presumably the same environment growing up.
2 totally different lives.
Oh.
It forces you to ask, what else is there? The researchers concluded that non-genetic factors had to be at play.
The software was running differently in each twin.
Exactly. One had the disease program activated. The other managed to keep it suppressed, despite having the broken hardware.
It really highlights that having the gene for something isn't the same as having the disease.
No.
But the source goes deeper than just random chance. It gets into something called imprinting. And this is where my brain started to hurt a little, so you're going to have to walk me through this.
It gets intricate, yes. but it's fascinating.
Okay, so usually we learn that you get one copy of a gene from mom, one from dad, and they both work together. If one is broken, the other one sort of picks up the slack.
That's the standard Mendelian model, dominant recessive genes. But imprinting completely breaks that rule.
Oh.
With imprinted genes, the cell actually knows which parent the gene came from. Some genes are chemically marked or stamped to be turned off strictly because they came from the father or strictly because they came from the mother.
The example they give, Prader-Willi syndrome versus Angelman syndrome, is incredible. I've heard of them as separate conditions, but I never realized they were two sides of the same coin.
They're often called sister syndromes for this reason. So look at chromosome 15. Specifically a region called 15q11q13.
Catchy name.
Very catchy. But it's a critical region. If there's a deletion there, a chunk of code missing, you get a disease.
Okay.
But here's the kicker. Which disease you get depends entirely on the parent of origin.
So let's play this out. Say I inherit this chromosome 15 with a deletion from my father.
If the deletion comes from the dad, you develop Prader-Willi syndrome. The symptoms are very distinct. Severe obesity, short stature, and this intense, uncontrollable hyperphagia.
Hyperphagia is the obsession with eating, right? They never feel full.
Yes. It's a relentless biological drive to consume food, plus developmental delays.
And why?
Because the genes in that region that are normally active on the father's copy are missing. And because of imprinting, the mother's copy of those specific genes is naturally turned off. So you have 0 working copies.
Okay. But now let's flip it. Say I inherit the exact same deletion in the exact same spot, but this time it comes from my mother.
Then you get Angelman syndrome. And the presentation is radically different.
How different?
These children often have a very happy disposition. They smile and laugh frequently, but they suffer from severe intellectual disability, speech impairment, and balance issues.
That is just, it's the aha moment for understanding complexity, right? Say missing hardware. But because of the parent of origin effect, the software crash looks completely different.
Precisely. In Angelman, you're missing a gene called UBE3A, which is only active on the maternal copy. The paternal copy is silenced. So if mom's copy is broken, you have no UBE3A.
It's so fragile. It all relies on this perfect dance of on and off switches depending on where the gene came from.
It is. And what's more, the source talks about how the environment, things that happen to us, or even to our parents, can reach in and fiddle with those switches.
This is the nature versus nurture debate, but it sounds like nurture is actually changing the nature.
That is a great way to put it. We used to think they were separate buckets. Now we know nurture spills into the nature bucket.
The source details this famous study on rats, the licking and grooming study.
This is one of the most cited studies in epigenetics because it implies agency. So researchers looked at rat mothers. Some were naturally very attentive, lots of licking and grooming of their pups.
And others were low-care mothers.
Right. And they tracked the pups into adulthood. They found that the pups who were highly licked and groomed grew up to handle stress much better. They were calm.
They were chill rats.
They were chill rats. The pups who were ignored were anxious and fearful. But here is the science. When they looked at their brains, specifically the hippocampus, they found the mother's behavior had physically altered the chemical markers on the DNA.
What? How?
Specifically, it changed the glucocorticoid receptor gene.
So the mother's care removed a block on that gene.
Exactly. It decreased DNA methylation is usually a stop sign, and increased histone acetylation, which is a go sign.
So the gene could be expressed more.
Yes, creating more receptors to soak up stress hormones. The software was reprogrammed by the mother's tongue.
That's incredible. And just to be clear, this wasn't genetic, right? If they took a pup from a bad mom and gave it to a good mom.
The pup turned out chill. Yes, it was purely environmental. And the source briefly mentions another example, the agouti viable yellow mice. In that case, just changing the mother's diet, giving her folic acid, changed the coat color of the babies and protected them from obesity and diabetes.
Just by changing the diet?
Just diet. It methylated the agouti gene and turned it off.
So we are not just building a Lego tower according to instructions.
We are constantly remodeling based on what we eat, how we're treated, and maybe even what our parents experience.
Okay, so that's the software, the epigenetics. The switches are dynamic. But before we get too carried away with the idea that everything is fluid, we have to acknowledge that there's a hard code underneath.
Right.
We wouldn't know any of this if we couldn't read the letters in the first place.
Correct. We need to talk about the hardware.
Yeah.
And that brings us to our second source, which focuses on Sanger sequencing.
Now, when I hear sequencing, I think of the Human Genome Project, you know, billion-dollar labs, AI, massive computing power. But Sanger sounds a bit vintage.
It is vintage. It was developed by Frederick Sanger back in 1977.
1977, that's almost 50 years ago. That's like the Atari era of genetics.
It is, but, and this is crucial, it was the dominant method for 40 years. It is still considered the gold standard.
Why is it still the gold standard if we have newer, faster stuff like next-generation sequencing?
Accuracy.
Yeah.
Pure and simple. Next-gen sequencing is fast. It's like skimming a book. You get the gist, but you might misread a word here and there.
Okay.
Sanger sequencing is like reading with a magnifying glass. It has 99.99% accuracy.
That high?
Oh, yeah. When you absolutely need to know if a specific letter is an A or a T, you use Sanger.
So how does it actually work? The source mentions chain termination, which sounds a bit Terminator-esque.
It's a clever bit of chemistry. I like to use Lego analogy.
On Legos, I can do that.
Imagine you're building a Lego tower. You have a bucket of normal bricks, red, blue, yellow, green. Those are your normal nucleotides, A, C, T, G.
Got it. Building the tower, just clicking them together.
But you also sneak in some special stopper bricks. These are the DDoxynucleotides, or DDNTPs.
Stopper.
Right, these bricks are chemically modified so that once you snap one onto the tower, you cannot add any more bricks.
The mechanism is jammed, the tower stops growing.
Exactly.
Okay, so I'm building this DNA tower, and suddenly, click, I hit a stopper, and I'm done.
Right. Now imagine doing this millions of times in a test tube. You end up with millions of towers of every possible length. Some stop at brick 3, some at brick 10, some at brick 500.
And how does that tell us the sequence?
Because the stopper bricks are color-coded with fluorescent tags. Maybe the A stopper is green and the C stopper is blue. So you line up all these towers by height, from shortest to tallest. The shortest one ends in a green brick. The first letter is A. The next one ends in blue. Second letter is C.
That is brilliant. You're reading the sequence by seeing where the construction got halted.
Exactly.
Yeah.
And while it can't sequence a whole human genome in an hour like the new machines,
It would take years.
it would take years. But it produces long, uninterrupted reads of about 500 to 1,000 letters.
And the source mentions that this long read capability is why it's not obsolete. It talks about viruses.
Yes. For example, the CDC's CalisaNet. When there's a norovirus outbreak, the classic food poisoning on a cruise ship.
The buffet nightmare.
The buffet nightmare. To track exactly where that virus came from, they need a fingerprint. They use Sanger sequencing to read the specific viral genes.
Because it gives them a long, clear read.
A long, clear read. They can see minute differences between strains.
And even more recently, SARS-CoV-2.
Right. When tracking variants of the virus, specifically changes in the spike protein gene, the S gene, Sanger is often used. That gene is about the perfect length for a Sanger read.
So it's distinct and accurate.
Very. So even though we're in this era of big data genomics, when we need to verify a specific mutation like, is this Omicron or Delta, we often rely on Frederick Sanger's 1977 chemistry to be sure.
So we have the precision tool Sanger to read the letters. We have the understanding of the switch's epigenetics to see how those letters are expressed. But here's where it gets really interesting. We have a third source today that suggests we might be able to skip the DNA test entirely.
This brings us to the paper titled Comparative Performance of Machine Learning Algorithms for Early Genetic Disorder Classification. And this represents a massive shift in thinking.
Huge. Because normally, to diagnose a genetic disorder, the logic is linear. You see symptoms, you suspect a genetic cause.
You order a genetic test.
You order a genetic test, maybe Sanger, and you get an answer. Or, sadly, you wait for symptoms to show up, which can take years.
Exactly. And that waiting game is dangerous. Many genetic disorders, like metabolic conditions, do irreversible damage before they're diagnosed.
And the tests are expensive.
Very expensive. It requires high-tech labs. In a developing country or a rural clinic, you might not have a sequencer. You might not even have a freezer to store the sample.
So what did these researchers do? They didn't feed the AI genetic code. They didn't give it the A's, C's, T's, and G's.
No, they didn't. They fed it what they call basic clinical indicators. Stuff you can measure at birth or in infancy with almost zero technology. We're talking about 42 different features.
Give me some examples. What counts as a feature here?
Things like white blood cell count, respiratory rate, heart rate, whether there was birth, asphyxia, trouble breathing at birth.
So basic medical chart stuff?
Very basic. Maternal age, family history, the stuff a nurse writes down in the first 10 minutes.
And they took a data set of over 22,000 instances and just asked, what? Can a machine look at these ripples in the pond and predict if the child has a genetic disorder?
That's the question. And not just yes or no, they were classifying them.
Right.
They tried to predict the class of disordered mitochondrial, multifactorial, or single gene. And then they went deeper, trying to predict the specific subclass things like cystic fibrosis, Tay-Sachs, or susceptibility to cancer or Alzheimer's.
So, drum roll. How did the AI do? Because this sounds like finding a needle in a haystack by looking at the shape of the haystack.
They tested a bunch of algorithms, random forest, gradient boosting, SVM.
The usual suspects.
The usual suspects. And the winner for the broad classification was an algorithm called Cat Boost.
Cat Boost. Sounds like a brand of energy drink for pets.
It stands for categorical boosting. It's particularly good at handling categorical data variables that aren't just numbers, but categories like high, medium, low, or yes, no.
And the accuracy.
It achieved an accuracy of 77% in predicting the class of genetic disorder.
77%. Okay, so let's be real. It's not 99.99% like Sanger.
No.
But for a screening tool that uses zero DNA, that seems significant.
It is significant. Think about it as a triage tool. If you have 100 babies and you can flag the 20 who are at high risk with 77% accuracy, just by looking at their heart rate and blood count.
You've just saved a lot of lives and resources.
You know exactly who needs the expensive DNA test.
And when they looked at predicting specific subtypes, it got even better.
Yes. For identifying specific conditions, another algorithm called support vector machine, or SVM, hit 80% accuracy.
80% accuracy for identifying things like cystic fibrosis just from clinical data. That implies there are patterns in our basic physiology that we humans are completely missing.
That's the black box of AI. It's seeing correlations that are not obvious to the human eye.
Right.
They did some clever feature engineering to help it along. They combined variables. For example, they created a feature called maternal age above 40, or combined heart and respiratory issues into a single flag.
So the AI might see that mother over 40 plus elevated respiratory rate plus low white blood cell count equals high risk for mitochondrial disorder.
Exactly. A doctor might look at those three things separately and say it looks within the normal range. The AI looks at the combination, the constellation of data points, and says, warning.
It's spotting the subtle systemic stress that the genetic error causes even before the full disease shows up.
Yes. And that matters so much because it's about accessibility. Democratization. Imagine a rural clinic in a remote area. No sequencer, no connection to a high-tech lab. But they have a computer. If they can input these basic numbers into a model and get a high-risk alert,
they can prioritize that child for transport or specialized care immediately. It closes the gap.
It does.
It's connecting the dots in a really beautiful way. We started with the hardware Sanger finding the needle. We moved to the software epigenetics, realizing the needle might be fine, but the instructions are wrong, like in the twins.
Right. Or in Beckwith-Wiedemann syndrome, which is another imprinting disorder. It highlights that the error isn't always in the letter, but in the instruction.
And now we are seeing that the errors in the hardware and software create ripples. And those ripples, like heart rate and blood cells, can be read by a computer to work backwards to the cause.
That's a beautiful synthesis. We used to think we had to find the broken gene first. That's the Sanger approach. But this machine learning paper is saying the haystack looks different when there's a needle inside.
But 77% to 80%? We still have a way to go, right?
We do. The paper explicitly mentions that limitation. They need larger data sets. The more data you feed these algorithms, the smarter they get.
So they're hitting a ceiling.
For now. It's good for screening, but not for final diagnosis. You'd still want to confirm with something like Sanger.
So they complement each other. The AI raises the red flag, and then you go in with the Sanger sequencing to read the code.
Exactly. It's a multi-layered defense system.
Yeah.
It's not about replacing the biologist with the robot. It's about giving the biologist a better map of where to look.
It's fascinating how these three sources talk to each other. You have the rigidity of Sanger finding the exact letter. You have the fluidity of epigenetics showing how context changes everything. And then you have the pattern recognition of AI showing us that the body is a system, not just a list of parts.
It reflects the complexity of biology itself. We are not just a code. We are a code plus a history, plus an environment, expressing itself as a living, breathing system. Genetic destiny is a myth because the system has so many entry points for change.
So as we wrap up this deep dive, what's the one thing you want the listener to walk away with?
I think it's that we need to stop looking for simple answers in biology. Between the epigenetic switches that respond to our mother's care or our diet, and the complex interplay of factors that AI is just starting to unravel, the picture is much more fluid than we thought.
Right.
We aren't just building a Lego tower according to instructions. We are constantly remodeling it while we live in it.
I love that. And I have a final thought to leave you with, something to mull over. We talked about how the AI could predict genetic disorders using basic clinical indicators like heart rate and white blood cell count.
Yeah, things we've been measuring for 100 years.
Right. It makes me wonder, what other hidden signals is our body broadcasting right now that we are completely ignoring? If an AI can diagnose a genetic flaw without ever looking at a gene, are we approaching a future where the phenotype What we see on the outside is just as computable as the genotype. Are we walking around with the answers written on our faces or encoded in the tiny fluctuations of our pulse, and we just haven't built the algorithm to read it yet?
That is a provocative thought. The body as a broadcast signal, waiting to be decoded.
Something to think about next time you check your smartwatch. Thanks for listening to The Deep Dive. Stay curious about the code and the context of life.
See